Hello, my name is Janessa Mendoza, and today I will be talking about a scientific research article named Protein Structure and Sequence Reanalysis of 2019 Novel Coronavirus Genome Refutes Snakes as its intermediate host and the unique similarity between its spike protein insertions and HIV. The bioinformatics method that I will be covering is whole genome alignment, and my audience is BIMM143 students. I'm going to start off with an introduction to the article. There are two main questions that the researchers in this article are investigating. The first question is, what are the possible intermediate hosts of the 2019 novel coronavirus, and how do the viral protein structures determine the identity of the 2019 novel coronavirus host? The second is, are the four insertions found in 2019 novel coronavirus and HIV-1 spike proteins an indicator of a unique relationship between the two that could help us make conclusions about 2019 novel coronavirus's evolution? The hypothesis is if snakes are not the intermediate hosts of 2019 novel coronavirus and there is no significance that the four insertions found in 2019 novel coronavirus are uniquely related to HIV-1, then there must be another intermediate host with relevant genetic similarities to 2019 novel coronavirus that could help us make conclusions about its evolution. So to continue going on with introducing this article, there are a few key terms to go over. The first one is CI-TASR, which is Contact-Guided Iterative Threading Assembly Refinement. This is an extension of ITASR used to create accurate predictions and models of protein structures. The next is spike glycoproteins. These are proteins that protrude from the outside surface of SARS-CoV-2 virions, and they help facilitate the virion's entry. The next key term is metagenomics, and this is the study of groups of genomes from a community of organisms, with these organisms usually being microbial. Next is muscle, which is also multiple sequence alignment comparison by log expectation. This creates alignments for multiple protein sequences in a quick and efficient manner. My last key term is an open reading frame, which is part of a DNA sequence that usually has no stop codons when it's translated into amino acids. These regions are usually studied since long open reading frames can be evidence of a gene. Now I'm going to go over why this is important. There are three take-home findings from this article. The first take-home finding from this article is that there is a close relation between 2019 novel coronavirus and bat coronavirus, in addition to refuting previous claims that show a unique relationship between COVID spike proteins and HIV spike proteins. The next take-home finding is that after performing RSCU analysis on many vertebrates, snakes with an RSCU distance of about 34.44 and 34.48 are not an intermediate host of transmitting novel coronavirus due to other vertebrates, for example, frogs with an RSCU distance of about 32.64 having a smaller RSCU distance. Additionally, RSCU analysis is an incomplete method in identifying intermediate hosts of coronaviruses due to lack of specificity to differentiate potential hosts. The last take-home finding is that malign pangolins are a likely host of 2019 novel coronavirus due to its high sequence identity of about 91% between Manus javanica and COVID spike protein and minimal residue difference. So in terms of what has been done before, on the first article on this slide, it was found that 2019 novel coronavirus may have originated from bats due to evidence of high sequence identity. So this helped the researchers investigate the possibility of an intermediate host. So in the next article, it argued that snakes are the intermediate host of 2019 novel coronavirus based of RSCU analysis on 8 vertebrates. In our article of focus, the researchers revisited that conclusion and performed a more encompassing RSCU analysis. In the last article that we are referencing, it claims that 4 new insertion sequences in 2019 novel coronavirus are only shared with HIV-1. From our article, a blast was done to see if the 4 insertions could be identified anywhere else. So our results from this paper are different from the results of previous work in a few ways. So referencing the first article, In this paper, building off of the evidence that bats may have been the original host, intermediate hosts were investigated. Next, our article found that snakes are not the intermediate host and found that RSU analysis alone cannot separate coronaviruses from varying animal hosts. Lastly, after conducting blast searches on the 4 novel insertions in our article, they found that they are not unique to HIV-1 and can be found in other viruses. So in terms of limits of applicability, there is limited experimental and clinical data that can be used to make statistically significant claims about the pangolin being an intermediate host. There's also an over-reliance to modeling and computational analysis to study the virus and lack of follow-up experimental data, so there should be increased caution to avoid overinterpreting any of the data. The bioinformatics method I will be covering once again from this article is a whole genome alignment. So here I have figure 5a, which shows the alignment between the human 2019 novel coronavirus full genome and the draft genome for malign pangolin manus coronavirus. So here on the bottom, the thin black line is the 2019 novel coronavirus full genome. And then going to the top thick black line are regions of the draft genome of manus coronavirus that are aligned, which means the two show genetic similarity. And looking at the thick black lines, we can see it should cover about 73% of the 2019 novel coronavirus genome, and it has 91% sequence identity. And this just means that 91% of these alignments had matched exactly between the two sequences. We also see little gaps in the top thick black line. These are just missing nucleotides of the manus coronavirus draft genome, and the way it can be improved is if it can be isolated in vitro and whole genome sequenced. The blue arrows represent open reading frames or protein coding genes. Again, open reading frames are sections of DNA that do not contain a stop codon. This figure helps support the author's hypothesis by identifying malign pangolins as a possible intermediate or even natural host. Here, an additional method was utilized to show that there was high sequence identity between manus coronavirus and 2019 novel coronavirus, despite spike proteins usually having to be more diverse to survive over time when they go from host to host. I'll now be going over some sample code. So step 1 is downloading the SARS coronavirus full genome as a FASTA file from the NCBI database and putting it into R. If not downloaded already, you will have to download the seqner package by using the function install.packages. The library function calls the seqner package to load, and then you can use the read.fasta function to read the FASTA file the sequence in, and I assigned it here to a variable COVID genome. Next is downloading the manus javanica, aka the malign pangolin genome as a FASTA file, also from the NCBI database, and putting it into R. So this is done in the same exact way that we read in the previous COVID genome, and here I assigned it to variable manus javanica. Next, we're going to go ahead and prepare the pairwise sequence alignment. If not done so already, you can install the package biostrings, and within biostrings, you can use the data function in R to load a data set. And here I use Blossom62, and it's a matrix that comes with the R biostrings package. Lastly, you can perform and print the pairwise sequence alignment. Here I assigned it to a variable genome alignment, and I use the function pairwise alignment. I made sure to put in the COVID genome, the manus javanica genome that we had uploaded, and for the matrix, again, I put Blossom62, set gap opening to -2, gap extension -8, and score only to false. For me, I did run into an error, and it said that it was unable to find an inherited method for function seq type for signature seq FASTA DNA. But here, this is what it would have looked like if the global pairwise alignment had worked. It would align them, and it would show their alignment score. Lastly, I just wanted to end with some questions. The first question is, does the diversity of bat species in 2019 novel coronavirus have anything to do with the diversity of potential hosts? Next, for MERS, another disease in the coronavirus family, could there also be another intermediate host playing a role from camels to humans? Thank you for watching.